Hello there everyone, welcome to Saurabh's classes. This is Ashmita. Here we are going to solve 5 to 6 questions based on the biotechnology from the IIT JAM examination for 2023. As you can see, I'm going to solve section A. I have chosen 5 to 6 questions from here and these questions carry 2 marks each. So let's start with question 11. Now the questions might not match with the sets that you might have got. So question numbers might not match, but the questions are quite similar. So similar, not quite similar, but exactly similar. So just check the questions and the answers. Which one of the following statements about G1 checkpoint of eukaryotic cell division cycle is incorrect? Which is not correct among them? Let's go through the options. Cell assures the existence of favorable extracurricular environment. This is okay. Cell assures the DNA uh, has no damage. This is also okay. These are the functions. Third one is also okay. Fourth one, yes. Fourth one is incorrect. Cell assures complete replication of DNA. Is an incorrect statement about the G1 checkpoint of the eukaryotic cell division. Okay, so this is incorrect. Talking about the G1 checkpoint, let's talk about this one. G1 checkpoint, this is also known as the restriction point. And this is that point in the eukaryotic cell cycle where the cell decides whether to proceed with the DNA synthesis, that is the S phase, and proceed with the cell division, or whether they will exit the cell cycle into a non-dividing state. Okay, that is the GO phase, we call it. The non-dividing state is the GO phase. Now, the G1 checkpoint ensures that the cell has favorable extracellular conditions. The DNA is undamaged, then it's mentioned in the second point that it sees to it that whether there is no damage in the DNA and that there is sufficient nutrient or sufficient nutrients to support the cell division. Okay. At the G1 checkpoint, what happens? The cell assesses the integrity of the DNA. Okay, and also detects any DNA damage. Sees to it that there is no damage, as it is mentioned in the topic only. Now, suppose if there is damage in the DNA, DNA has damage. If DNA has damage, what is done? Okay. If DNA damage is detected, the cell activates the DNA repair mechanisms, okay, before proceeding with the cell divisions. This ensures that the DNA is replicated accurately and that the genetic information is preserved. Okay. Therefore, we can say that the most incorrect one is option D. Okay. Let's move over to the next question. We have question 12 next, and this is based on reasoning and assertion. Determine the correctness or otherwise of the following assertion A and reason R. Assertion says nitric oxide is involved in transient paracrine and autocrine signaling. Reason is nitric oxide is highly reactive with a lifetime of few seconds, yet can diffuse freely across membranes. Both are true, and reason is the correct for assertion. That's the first point. I think the first one is going quite correctly. Both A and R are true. Yes, actually, whatever is written, it's true only. Nothing problematic. Let's go with option A. I don't think we should consider any other points over here. Go with option A. Both are true and the correct reason for A is also there. In correct reason. So nitric oxide, let's discuss about this now. Nitric oxide or NO is a signaling molecule that is involved in many physiological and in many pathological processes in our body. Now, it acts as a transient paracrine and autocrine signal by diffusing freely across the cell membranes. and also by interacting with intracellular targets. Okay. Now the assertion that is given to us, that is nitric oxide is involved in transient paracrine and autocrine signaling. Obviously this is true because right now we have discussed nitric oxide is produced by various cells in response to the various stimuli and acts that locally takes place on the nearby cells to regulate the cellular functions. And along with that, let's come to the reasoning part. Nitric oxide is highly reactive with a lifetime of few seconds, yet can diffuse freely across membranes. This is also true, as I have already chosen the correct option. This is also true, and it also provides a correct explanation for the assertion, obviously. So NO, we can consider NO, a nitric oxide, we can consider it's highly reactive molecule, okay, as it is written over here. It's highly reactive molecule and has a short half-life of only a few seconds. However, its ability to diffuse freely across the cell membranes allows it to rapidly and efficiently interact with the intracellular targets, making it an effective signaling molecule. So yeah, option A would be the correct answer. Let's move over to the next question. This says, which one of the following statements is correct about solute transport across membranes? Let's go through the options once. Passive transporters decrease the activation energy and does not facilitate the transfer of polar compounds. Um, no, not really. Second one says direction in which a charged solute tends to move spontaneously across a membrane does not depend on the electrical gradient across the membrane. No, this is also not going. All the ABC transporters do not have nucleotide binding domain. This is also not correct. P-type ATPases get reversibly phosphorylated as a part of transport cycle. Yes, this one is correct. They get reversibly phosphorylated as a part of the transport cycle is the correct statement about solute transport across the membranes. Okay. Now talking about solute transport. What is this all about? Solute transport across membranes actually occurs through either passive or through active transport mechanisms. Okay. Passive transporters. Let's talk about passive transporters now. Passive transporters facilitate the movement of solute down their concentration gradient without the expenditure of any kind of energy. Now, active transporters, what do they do? Active transporters, on the other hand, they require the expenditure of energy to move the solutes against their concentration gradient. Okay. So from this discussion, we can come to the conclusion that option A is incorrect because the passive transporters do not decrease the activation energy. They simply facilitate the movement of solutes down their concentration gradient. Okay, and these passive transporters can transport both the polar and the non-polar compounds depending on the nature of the transporter and the solute. Okay, that is why this is incorrect. Now talking about the second option. Option B is incorrect because the direction in which a charged solute is or tends to move spontaneously across the membrane, that depends on the electrical gradient across the membrane, okay, as well as the concentration gradient of the solute. So that is why option B is also incorrect. Talking about option C, all the ABC transporters do not have nucleotide binding domain. This one is also incorrect because all ABC transporters have nucleotide binding domains, of course, which are necessary for their function. So here, just the wrong thing is written. Let's talk about option D now. Now, Is in this is correct because the p-type ATPases, it's a type of active transporter. Like when I was talking about active transporter, this is an active transporter, and are like reversibly they are the phosphorylated during their transport cycle. They are reversibly phosphorylated during their transport cycle, these active transporters. So the phosphorylation and dephosphorylation of the specific amino acids reduces in the transporter protein, which provide the energy which is necessary for the solute transport. Okay, so that is why option D is correct for us. Let's move over to the next question. It says match the type of DNA repair mechanism in group 1 with the enzymes involved in group 2. We have here the alphabets as group 1 and the numbers as group 2. This is group 2 and this is group 1, right? Let's try to match them. Now the correct one would be mismatch repair. Mismatch repair P will go with option 4. Base excision repair will go with DNA glycosylase. Okay. Then nucleotide excision repair will go with, yeah, the second option. S or double strand break repair will go with the third one. So let's find out this combination. Where can we find it? It's given in option C. Yes. Option C, isn't it? Yes. Option C. Let's talk about the next question, that is in the southern blot technique. Which of the following reagents is used to detect the presence of a desired DNA fragment? Now, in the southern technique, we get to see a DNA probe. Okay, we get to see a DNA probe which is used to like detect the presence of a desired DNA fragment. Okay, that is why we are choosing DNA probe as our correct option. Let's talk about the last question for today's class, which is which one of the following statements about photoproteins in plants is incorrect? Which one is incorrect? Now, the incorrect one is phototrophins are insensitive to blue light. This is incorrect. Okay. Um, Now, phototrophins are actually sensitive to blue light. Okay, and they play a very, so we can say they are sensitive to blue light. They are sensitive, so they play a critical role in phototrophism. All right, that is the bending of the plant stem towards a source of blue light. Therefore, the correct answer would be the one that I have chosen. Okay. Phototropism. They play a major role in this. All right. Yeah, so that was it for today's class. If you want to have more detailed discussion in more detailed manner, if you want question answer sessions, then we can have those in our classes. We provide live lectures, doubt clearing sessions, mock tests, and any kind of exam course preparation is done over here. Our expert guidance will help you to crack all the examinations. And if you really want to prepare well with us, then you need to join our classes. For that, all the details are given in the description box below. Do give it a visit and join our classes so that we can just help you out with your exam preparation. Thank you for watching.